Hi, it's Mr. Mintner, and welcome to our genetic screencast. This is Unit 5 for my 7th graders at South Middle School in Henderson, Kentucky. I, if you like the background, we're going a little casual tonight. I'm in my living room, and I don't have my pad to draw on or anything. We're going to do it all with the mouse and see how we do it going casual tonight. Anyway, genetics all begins, the unit we're starting on all begins with this guy right here, Gregor Mendel. Gregor Mendel was a monk that worked with pea plants in his monastery in Austria in the 1860s. And he come up with all of the ideas. He basically bred peas for years, over 30,000 different pea plants he did experiments with to come up with all of the ideas we have for genetics. So he bred peas. Basically, he took this pea and said, "Hey, how you doing? There's this pea over here," and he got the peas together with like 85 levels of compatibility. It was kind of like eHarmony. It was like pea harmony. So he like got these things together and he bred them and he come up with all of this data that basically described all we know about genetics. Now here's the cool thing. Later on, we discovered after his death all of this stuff we talked about in the last unit about meiosis, about chromosomes. We didn't know anything about chromosomes, Mendel didn't, and that all of those genes that he was talking about in those peas were actually on chromosomes. Now, this is a human karyotype, which means I've got 23 pairs or 46 chromosomes. And so right here might be one of those genes he was talking about, and right here would be one of those genes. You got one of these from your mom and one of these from your dad, if you remember from before. And through the process of meiosis, where I start off with 4 here, I end up with 2 each. I have a haploid number. I split it in half. So going back here, I have 46 chromosomes, but whenever I make sperm or egg cells, gametes, they each get 23 chromosomes. So when they come back together, like right here, I end up with 23 in this one and 23 in the egg, and I end up with 46 total, so it looks back like a normal human karyotype with 46. All that's review from last time, but what I want you to know is that this new material and the old material, they go together. They're hand in hand. Just Mendel didn't know this stuff at the time. He was just working with the peas and he discovered all this stuff without knowing the other, which I think makes it even more impressive. John Lennon said it best later, and he said, "Hey man, all we are saying is give peas a chance." All right, so here we go. We're going to move on and look at the keywords for this unit. Trait is a keyword. What trait is it is whatever it is you're looking at, like eye color, hair color, whether your earlobes are attached, where your hairline is, does it come to a point like a widow's peak or straight across? All of those things are traits that are in your DNA that's inherited. So Mendel didn't know about DNA, but he called those things traits. Anything that was inherited was a trait. Alleles are the different forms of the trait. So I might have, if eye color is my trait, my alleles might be blue eyes and brown eyes. 2 different alleles for the traits. Okay? If I'm looking at cats, whether they have a tail or don't have a tail, the trait would be tails. The allele would be tail or no tail. Some cats actually are born without a tail. Okay? Dominant and recessive. What that is when Mendel was breeding plants, he said, if I got a tall plant and I put it with a short plant, all of the offspring become tall. Something happens to what made them short. It's not there anymore. So he called the thing that showed up dominant and the thing that didn't show up recessive. So that whenever they're together, the dominant always dominates over the recessive, so the recessive doesn't show up. The next 2 words are homozygous and heterozygous, and what that means is homozygous are when the alleles are exactly the same, and heterozygous are when they're different. So if I could write on here a little bit, homozygous would be like big R, and we usually have the capital letter for the dominant trait, and lowercase letters for recessive. Since they're both the same thing, they're homozygous. Homozygous dominant. However, if I did little r little r, this is working okay. That is homozygous because they're the same. Recessive, it's the recessive allele. Heterozygous, on the other hand, that is when I have two different alleles together. Big R, little r. Now in this case, they're heterozygous, they're different, but the trait that shows up is the dominant one. Whatever big R represents, that's the trait that shows up because it dominates over the recessive. Well, so let's just say big R represents red, all right, in something like maybe coat color in some kind of animal, fur color, and a little r represents white, all right? And so what happens is, if I am, okay, I'm trying to write with this, there we go. If I am homozygous for this, I am red. If I'm homozygous recessive, it's white. But if the two come together, it's not pink, it's red, because red is dominant over the white. Okay? Now, the last two words we want to talk about are genotype and phenotype. Phenotype, the PH, stands for physical characteristics. Another thing, that the word that starts with PH is photograph. If I were to take a photograph, what it looks like is the phenotype. So the phenotype here would be red. That's the phenotype. The genotype are what are the genes that makes this. So big R, little r is a genotype. And big R, little r are genotypes. They both are different genotypes, but the phenotype for both is red. Phenotype's what it looks like. The genotype are the letters. Little r would be a genotype that represents the phenotype of white. Okay? So our trait is coat color. The alleles would be red and white. Dominant, the red is dominant over white because when they're together, red is all that shows up. Homozygous are when the two alleles are the same. Heterozygous are when they're different. And then genotype are the letters, and the phenotype is what you actually see. Now, what we didn't know at the time, what he didn't know when he was coming up with all this, is that all of that is in here. Those genes are located on these chromosomes in the DNA. Okay, so those are the key words. Let's move on now to how we would solve problems, because this is going to be the big part of the test. Knowing these words and how to apply those would be one way that you're going to get points on the test, but a lot of the points are going to have to do with doing what's called the Punnett square. So let's take a look at that. Steps in solving Punnett square problems. Okay, here's how this works. If we identify the traits, first we're going to identify the traits and the alleles. All right. Then we're going to determine the genotypes of the parents. That's the most important part. That's where the people make the most mistakes, is trying to find what the genotypes of the parents are. Very important step. Then we're going to use the possible gametes of those genotypes to create a Punnett square, which I'm going to show you, and then we analyze our data. So those are the 4 steps in solving any Punnett square problem. So let's practice, okay? Here's a practice problem. Johnny Avatar from the movie, he's purebred for a tail, and he has a child with Susie Avatar, who is heterozygous with a tail. What is the probability they have a child without a tail? Well, what we first have to do is we have to identify the trait. The trait is they have a tail or no tail. It's about tails. That's the trait. And the alleles, if I go back to the alleles would be tail or no tail. Okay? So what I need to do next is determine what is dominant and what's recessive here. Having a tail or not having a tail. Usually the problem will tell you. This one's a little tricky because it doesn't. So you have to read between the lines. The fact that it says that this was heterozygous with a tail, the only way that you can have a recessive allele or recessive alleles show up in the phenotype or what it looks like is if they're homozygous. Because remember, if the dominant trait is there, that would mean it's heterozygous and the dominant one would show up. So if the heterozygous mean I got 2 different traits, which means that I got two different alleles for the trait. If the dominant allele is there, that's what shows up. So the fact that they have a tail and it's heterozygous means that tails, I'm going to do it like this, tails, I'm going to draw with a T, is dominant. So I'm going to do it with a capital letter. And little t would then be recessive, and that is for no tail. And you're going to say, hey, you're not writing very well, but if you've seen my writing in class, it's probably just as well. All right, so we've done that part. The next step is, if I were to go back, because I need to determine the genotypes, the genes of the parents. Johnny Avatar is purebred for a tail. That means he has a tail. Purebred means that he's homozygous. So Johnny, if I'm going to change my color. Johnny is going to be big T, big T. All right? Susie is heterozygous, so that always means I have two different alleles. So she's big T, little t. Okay? Now, here's what I want you to do. The next step is we draw the Punnett square. Now, Johnny, if he undergoes meiosis, he's going to split those alleles apart, and he can either give a big T or a big T. When those split, he can only give a big T, okay? He's going to make sperm that are haploid, remember, that only have half the chromosomes. So he can't give both of these T's. He can only give one or the other. But since they're both big T's, he can give a sperm with a big T, or he'll create another sperm with big T. That's all he can give. Susie, on the other hand, when she splits hers and makes eggs, she can give a big T or a little t. Now, what Punnett said, he came up with a way to show this, and he created what was called the Punnett square. And if I draw a Punnett square, I'm going to do it over here. It's like a square with a cross in the middle. And we're going to take the possible sperm, and we're going to put them up top. Big T, big T. What that is that's that big T and that big T. They come down here. But what I want you to understand is they represent possible gametes or sperm cells. The big T, little t for her, Susie, will put on the side big T, little t, and they represent possible egg cells that are haploid. She can only give one or the other. She got one of these from her dad and one from her mom, and she can only give one of those to her child. Johnny got the big T from dad and a big T from mom, and he can only give one of those for his child. So what this represents is if that sperm and that egg go together in this box, it shows that I have a big T and a big T. That is homozygous for the trait of having a tail. This one goes together. It is big T with that sperm and that egg coming together, big T, big T. And that is also homozygous for the trait of having a tail. This one right here would be big T, little t. That sperm and that egg coming together. That is heterozygous, because they're different, for having a tail. And if that sperm and that egg come together, we drop this down, big T, and this little t comes over. And that would be heterozygous for having a tail.